Hey, welcome back to the channel. This is Joel Duff, and today, quick episode of Critiquing Creationism. This is where we just take a look at a quote by young Earth creationists or a short article by young Earth creationists and ask ourselves, what are they trying to say here? What is their fundamental point? And potentially, what do they have wrong about this? What don't they understand? And so today's quote comes from Ken Ham, CEO of Answers in Genesis, who on the Answers in Genesis social media account was posted this particular quote. There is no known natural mechanism that can create brand new functioning genetic information. Okay, bunch of words in there, right? Naturalistic, creation, brand new, functioning, genetic, and information. All loaded terms that likely have definitions, right, that we need to define. And what is the definition of these terms to a young Earth creationist? Why do they think they can say this with great confidence? Because Georgia Purdom is going to be, who is the person who's quoted, well, actually, Ken Ham is quoted here, but it references an article to back this up, presumably with the evidence for this particular quote, because again, Ken Ham doesn't know anything about molecular genetics. And so they bring in their resident PhD molecular geneticist, Georgia Purdom, to back this particular claim up. So what's going on here? Is there no known natural mechanism that can create brand new genetic information? I think it's a nonsensical statement. I've provided many examples on this channel with many previous videos of how genetic information can be generated through a variety of different processes. But the reason I'm highlighting this particular quote and the response that Georgia Purdom has to it, or the explanation Georgia Purdom has to it, is that I think it will tell you and teach you fundamentally how young Earth creationists think differently, about information and data, and they have different definitions for these terms. So here's the article that is linked to that particular quote. Evidence of new genetic information, ? By Georgia Purdom. And this is, I said it was 2013. This is actually farther back, 2008. And she's responding to somebody who had feedback, which was, hey, there really are ways that genetic information can be made by naturalistic processes. And she says, no, it can't. And you see here, can duplication and mutations cause new information to arise in the genome? These are the two methods that she's going to respond to, although there are other mechanisms for creating genetic information. But let's just stick to these two, because that's what she's going to refer to. And this is all I'm here for, is to point out this quote here. As it has been said many times on this site, duplications and mutations do not add new information to the genome. And you may be sitting there thinking to yourself, wait a second, if you duplicate DNA, then you're adding more nucleotides to a genome. You're increasing its size. And especially if you duplicate a gene, then you would have two copies of a gene. And if you have two copies of a gene, well, they could be expressed differently, or you're going to make more of a product than you were making before. And doesn't that make the organism different? Haven't they added something new to what they previously could do if you've duplicated something? Also, potentially, if you duplicate a gene or duplicate a whole segment of DNA that includes multiple genes, and then you have multiple copies of that DNA, could not some of those copies of DNA have mutations and change the function of one of the copies of those genes? So therefore, you now have two genes doing two different functions, whereas before you had one. How could that possibly not be considered an increase in genetic information? I don't like the word information there, but we're going to use their word genetic information. How could that not be an increase in, I'll say, genetic capacity, the ability to do things? In other words, we're adding new features to organisms. If you add a new feature to an organism, aren't you adding new information? According to Georgia Purdom and Ken Ham, most other young Earth creationists, this does not count. This is not adding genetic information. And you can see there in the next sentence, duplications are the result of duplicating existing genetic information. And mutations, all they do is alter existing genetic information. Neither of them adds new genetic information. So now ask yourself the question, what did they mean by genetic information? They meant the original nucleotides, right? They meant the original construction of a genome, right? From non-nucleotides in order to having a sequence of nucleotides, right? To them, that is creating information, because before you had random bits of nucleotides, and then when you put them together into a string, you now have genetic information. And then everything after that, if you duplicate that, you haven't actually created new genetic information, because that was already information. You've simply made more. I guess I can't even say the word more information, because they're saying no new information can be created. You're not creating a new code like with new nucleotides or anything like that. You're just making more of what was already there. You see how fundamentally now this is not very interesting? Okay, it's like, so anytime you try to say, like, if you copy DNA, if you have mutations, if you have an enzyme that has mutations and it changes what the enzyme does, so now it creates a new gene from that particular same sequence or similar sequence, they're going to claim that's not new information. You already had the DNA and all you did was alter the information. It's okay to alter information. You haven't actually created new information. So they've compressed the word information down into, I'm having trouble explaining this because I don't fully understand like what they think, what in their minds. I can't sometimes get in the mind of some young creationists, despite how much time I've spent reading and discussing this with them. What is the original information? Now, I will acknowledge that they will say that the original information would be like what God created for a kind. All right, the canine kind was given an original genome. That's the original information. And then they'll say, I think what they're suggesting here is that any changes to that genome, they could duplicate parts. All right, they could have mutations that change regulatory elements, and so they turn genes on and off at different times. They could even create possibly new variations of enzymes from that. They would claim none of that counts as new information. You're just manipulating the thing that's already there. So, yeah. So apparently, to younger creationists, you could have a gene duplication, and the duplicated copy of the gene could take on a new function because of a mutation. And as a result of that new function, the organism could have a new feature, right? They have the capacity to do something they couldn't do before. Even just duplicating genes, as I said before, you then have two copies of something, which means you can use both of them to make products, which means potentially you could make more product, like more enzyme, which would speed up that particular chemical reaction that it's doing. Right. So that in itself is going to change potentially the development of the organism. The expression of some particular feature will be changed. But that's how we make giant watermelons and other types of plants, right? We've duplicated their whole set of chromosomes, so they have twice as many chromosomes. To me, that feels like that's a lot more genetic information. You have a lot more code, right? And you have multiple copies of genes. And as a result of all those multiple copies of genes, there's an effect in some plants where they create like extra large fruit and larger leaves and so forth. Doesn't happen so much in animals in terms of that effect, but in plants, that's something that is quite frequent. And so we sometimes intentionally cause plants to duplicate their genome in order to potentially get these benefits, at least the benefits to us, not necessarily to the plants. But for answers in Genesis and Ken Ham, they're going to say that's not adding new genetic information. You're already taking what was God given and you're just messing around with it. Now, of course, the question becomes, how much can you mess around with it? What's the extent you can mess around with it? If you added 1,000 new genes through duplication and the organism now is morphologically completely distinct and utterly unable to replicate, to reproduce with other members of the own kind, what stops them from becoming other kinds of things? What stops them from evolving into other types of organisms, even though they have no new genetic information, technically, according to the answers in Genesis? Oh, let me read this last part here, because I think this will maybe help sink in, like, what is Georgia Purdom thinking here? Because she tries to provide an analogy. Think about it this way. If I give someone a copy of a book they already own, then they don't have any new information. Okay, that's an interesting way of thinking about it, right? It's like, if you have a copy of a genome and then I give you another copy of that genome, you wouldn't have any new information. However, she's actually wrong about that, because if you have two copies of a gene instead of one, yes, it is the same gene making the same product, but if you make more of that product, in other words, if you express both of them where you only had one before, you will alter the physiology, all right, the expression, the state of that particular organism. This is a reason why there are certain portions of your chromosomes that if you have 3 copies instead of 2, like trisomy 21, Down syndrome, if you have 3 copies, do you have more information? According to Georgia Purdom, you do not have any new information. You have the same information. It's not new. And yet it has an effect on the organism. Creates a different set of characteristics, right? So how are you defining that as not new information? You just have a copy of the information they already had. If I subsequently take a marker and mark out some of the letters or words of the copy of the book I gave you, they still don't have any new information. It's just messed up copy of one of the books. See, this really gets down. That's why I love this. These 2 little paragraphs right here really get you to the heart of like what many young Earth creationists are thinking when they talk to you about genetic information and what can happen to DNA over time, right? It's just like, you've got copies, you have the original perfect copy. And if I blot out certain letters, mutations, in other words, if I make mutations, I'm just messing up what was already there. And she would say even a duplication is messing that up because it wasn't part of the original design. And now I've duplicated it. And even though I can make something else, apparently that's not part of the original plan. So therefore, even though it has changed the organism, it's apparently not changed it for the better. I don't know whether that needs to be, new information doesn't necessarily have to be good information, right? It's like, it's just creating new characteristics, new features for that organism. So at the end of the day, right, they can explain away everything, because they can simply say that any change you make, adding, subtracting DNA, altering the DNA, forming new copies, new genes, new copies of genes, changing what a gene does, changing a regulatory network, because now you have repetitive sequence, like maybe you have multiple copies of a repeat unit that then interrupts a regulatory element. And that as a result, you don't turn on a gene at one time, you turn on at a different time. And that changes the development of an organism, right? All of those things are changing organisms. They're allowing organisms to adapt and change in their environment. And anthropogenesis says that yes, God provided the ability for organisms to change so they can have this malleability. They can reshuffle their genes and so forth, and potentially even add new copies of genes. And they'll say like, well, that is not evolution. And that's not new genetic information. So let me push this a little bit. Let me be a little absurd. I've done this in the past, and I think it's worth bringing this up again, this particular example. Let's say I have a little circular genome, okay? And it's got, 1,000 genes, right? So it's like a bacteria, right? And then I take that circular genome and I have a duplication. And so now it has 10 extra copies of some of the genes, right? 10 genes get copied. So now I have 2 copies of each. And one of the copies stays the same, but the other one has a mutation in it. And now it has a slightly different enzymatic property, right? Maybe I can break down lactose, right? Or galactose instead of just glucose. And so as a result, in the environment I live, I now have a new food source. right? Before I could only eat sugar, glucose sugar, but now I can actually take in other forms of carbohydrates and I can break them down and get energy from them. I gain an advantage over my neighbors who don't have that. So that duplication of that gene or set of genes and having them have mutations results in me being able to do bioactivities that I couldn't do before. Now, according to Answers in Genesis, this is not new information, and this is not evolution. Right? It's adaptation, I guess, to the environment, but that organism hasn't evolved because it can't get new genetic information. But what if I duplicate the entire genome? Now I got 2 copies of that genome, and each copy of the genome does slightly different things, right? And then I add new copies of those genes, and eventually I've got 5,000 genes. Right, and I'm creating new regulatory networks, and I'm copying little sections of DNA that eventually the bacteria figures out how to do more complex regulatory pathways, and therefore they have more complex biochemistry, allowing them to control their environment even more than they could before, than their ancestors could. Right? So none of this is evolution, right? This is just adapting to the environment, but the organism is changing to the point where probably it's not the same kind of bacteria as it was before. I take an animal and it duplicates its genome, which some fish have done that, and now they have 4 copies of all their genes, and then 2 sets of those genes end up, some of them get lost, some of the genes get changed into other things. I've shown many examples right on videos in the past, and could provide many more examples of whole genome duplications that have resulted in radical changes in that organism over time, to the point where they're changing into things that they have capacities and can do things that their ancestors had no capacity to do. And then when we toss in horizontal gene transfer, where you take this fits Answers in Genesis as well. If you take a set of genes, an operon from a bacteria, and you move it into another organism, which has happened and does happen, right? And you move a set of genes that has a biosynthetic pathway into a different organism, and it then begins to utilize that biosynthetic pathway, then that organism now has something it didn't have before, right? Fundamentally, it can do a biochemical pathway that it could not do prior to that in its history. But according to Answers in Genesis, you know what the response to them is to that? The response to that is, that's not new genetic information. It might be new to that organism, but it's not new genetic information because it already existed in the world. That operon already existed in another organism. Now, maybe that operon was created by 3 genes that copy themselves into 6, and then the three of them took on slightly new functions, slightly different versions of those enzymes, creating a more complex biosynthetic pathway. Then we took those 6 and we transferred into another organism, and now they benefit from that. Even that is not the creation of new genetic information, according to Georgia Purdom and Ken Ham. They're going to say this quote still applies. There's no naturalistic method that can create brand new functioning genetic information. You get that? Now I come back to the brand new word. The brand new is there's nothing brand new about a gene that's copied. It's copying something that already existed. And if you have a mutation, you're just changing it from one base to another base of, something that you're just changing the information there. It's not brand new information. Brand new information would be like, there's 10,000 base pairs that didn't exist in any organism anytime. Suddenly they appear and they have new features. And now the organism has something that it didn't have before and that didn't exist before. That would be new information. In other words, miracles, right? They're saying because you can't show that there's miraculous new DNA that suddenly appears in organisms, therefore there isn't any mechanism. Well, of course, what I just said doesn't make any sense because they're saying there's no known natural mechanism. They're saying there's only one mechanism, and that is a miracle. Except they don't expect that to happen, right? I don't think they think that, new DNA is popping up in any organisms right now. Right, so I said, it actually doesn't sound that absurd. I'm just trying to push the creationists back to say, how far back do you have to go? Right? Could I start with 1,000 base pairs of sequence and say right, There it is. There's 1,000 base pairs of sequence. I don't know where it came from. Maybe that came from a creator. God started with 1,000 base pairs of DNA. And then that 1,000 base pairs had mutations and duplications and grew in size into genomes that are 3 billion base pairs in size now. I mean, some are 100 billion base pairs in size, but us, 3 billion base pairs in size. And that 3 billion base pairs ultimately is just a copy of an original 1,000 base pairs. And since it's only a copy of what was already there, right, what already existed, none of that is new genetic information. It's not brand new genetic information. It's just a reformulation of a pre-existing thing. Right? See, Cunningham and Georgia Purdom, they know there's natural mechanisms that can create and form new genes. Right? But because they're formed from prior DNA, they're going to use these types of statements that make it sound like there's no way that new things can be made in organisms. But you get underneath the hood and it's kind of like, oh yeah, there can be mutations. There can be a horizontal transfer, right? There can be gene duplications that can affect organisms and actually add new characteristics to those organisms. We're just not going to call that new genetic information. All right, I'll toss one last thing in, and it's something I've mentioned as well in the past. I've even done a little experiment to show this is the case. If you were to take, because one other way to make new information in what I think is a better definition of information, which is just any new, any expressed DNA that does some kind of, performs some sort of function, right? It doesn't have to make a protein, right? There's little pieces of DNA that control other pieces of DNA, right? The transcription elements and so forth, right? That control when you turn genes on and off, right? And so if you create a new one of those, that's going to affect how other genes are turned on and off, which changes the effects of organisms. So there is another way of forming that kind of information, and that's called de novo creation, which sounds like from nothing, right? It's like, but in a way it's properly named because it's from random sequence, like sequence that doesn't have any necessary function. It's just A, T, C's, and G's. So I've done the experiment. I've had my students actually do this. You just say like, I would randomly generate 5,000 A, T, C's, and G's. All right. And since you're randomly generating, you could have a computer do it. I could do it too, but let's say you just have a computer generate A, T, C, G's in a random sequence. You can't say that although they were made and they were designed in a way, it wasn't designed to do anything. All right. That's what you think of as like sequence that doesn't have any function. It's just sort of like it's there. And I said, now take those 5,000 base pairs and look and see if you can find any genes or places where you could express or transcribe the sequence. And sure enough, out of 5,000 base pairs, even if you just randomly write it out, you're going to have certain short stretches that are going to be transcription initiation factor sites, all right? Promoters. Little short sequences where your proteins bind to that DNA and it tells the machine to copy that sequence and make it into an RNA. And there's going to be ATGs. ATG, just that 3 base pairs, that is the start codon. That tells the ribosome that's reading RNA, hey, there's a start codon right here. I can start reading the code. And if it can find an ATG, which out of 5,000 ATCs and Gs written out randomly, there's going to be dozens of ATGs, which means there are dozens of start codons. And then after that, there's going to be a series of 3 base pairs, 3 base pair, 3 base pair, 3 base pairs. And some places there's going to be a stop codon by chance in your randomly generated sequence. And therefore, you can predict that out of the random sequence you made, there are places that a organism would try to copy and read the code and potentially make a small little piece of a stretch of amino acids, a polypeptide, right? A protein. Now, you might say, well, that protein doesn't do anything, right? But it does get transcribed and made into a protein, right? So it's actually doing an activity in the cell. And you can prove that this happens, right? Because you can generate 5,000 base pairs and actually make the 5,000 base pairs in real, like actual real code, ATCs and Gs. Shouldn't say code. Real nucleotide sequence, ATCs and Gs. And then you can splice that into an organism, and then you can watch and see what the organism does. And you can see the organism will actually try to read that code and turn it into a potential protein, or at least it will make it into RNAs in the cell. So I could generate random sequence that I would give to a young earth creationist, and they would say that is functional based on some of their definitions of what makes DNA functional, right? They would look at that and say, oh, that's functional DNA. Now, I would say that most of that's not actually functional. Right? If it's not actually benefiting, or if it's not actually causing a real effect on that organism. But if you write out millions of pieces of sequence and you allow an organism to look at it, they're going to find some proteins that are probably going to have some use to them. In which case you've added de novo, brand new DNA that didn't exist before to an organism, and it can potentially find a use for that. I would think that that would be brand new functioning genetic information. All right, to sum up, so is there no known natural mechanism that can create brand new functioning genetic information? I'd say absolutely not. There's many, many, many examples of it. But if you follow the definition, right, if you accept the definition of genetic information, right, or new genetic information that Georgia Purdom provides to young earth creationists, then I guess you could say that this is an accurate statement given their definitions. But they're not really talking to an audience that understands their definitions, right? They really aren't, right? They just read, oh, there isn't any natural mechanism. They don't think that there's any mechanisms for copying and duplicating and changing DNA, when in fact there are. They're simply saying that doesn't count. They're defining it away, right? They're just saying like, nope, none of that counts. So whatever I would bring, they would just say, well, that DNA already existed. So therefore, that's not actually new genetic information. I could substantially change an organism. I could change an organism to something completely different by simply by copying parts and changing parts of that organism, and change it into a very different organism. And they would still say, you didn't create any new genetic information. You just swap parts around. But if you're going to say that, then, then you can't say that cats and dogs don't have a common ancestor. There could be a common ancestor organism that then swapped and moved parts around, right? Had mutations, shuffled things, had duplications, changed regulatory elements, and you end up with cats and dogs, right? I mean, are there any cats and dogs do have different numbers of genes. Cats have genes that dogs don't have, right? But some cats have genes that other cats don't have, and dogs have genes that other dogs don't have. I mean, every species has that kind of variation, yet young earth creationists accept that those species share a common ancestor, right? And they would just use this, they're reshuffling, they're having mutations, they're losing genes, they may be duplicating genes, right? There's clearly duplications in some species. But they would say, nope, none of that counts as new genetic information. You're not really adding anything new to the organism. Well, I could take an ancestor to a carnivore, and without adding anything new, I could make every type of carnivore using their understanding of what doesn't count as new. So it's kind of a useless definition because it doesn't help them define anything other than maybe you can't explain the origin of life, the origin of the original DNA strand, right? Because that's where the conversation will go. If you push it, I know that's where it'll go, because if I say, well, I could do this, well, you can explain the original sequence, right? That's what it'll always come back to, is that when I say, okay, so you could start with an original bacteria and you could make all of life on Earth, and none of that is new information, right? Right? They're all natural mechanisms, but none of them created new information. So therefore, natural mechanisms can create the entire diversity of life if you give it an original molecule. Yeah. And that sounds kind of silly to talk that way, but that's where young earth creationists have kind of painted themselves in by their wacky definition of genetic information. All right, that's it. I said this would be really short, and let's see, where are we at here? 30 minutes. All right, I'll try to cut a little bit out. Great talking to you. We'll see you later. Bye-bye.